Good morning, friends. So, in this lecture, we are going to see some of the common terms that are being used in the animal genetics. So, this is the beginning of Unit 2 of your syllabus. So, at the beginning, we should know what are the terms that we are going to use in this unit frequently. So, few of the terms we are already knowing, and there may be one or two terms that you might not have been heard. So, just we will refresh that what are the terms that are being used in animal genetics. So, first of all, the most common term is DNA. So, what is DNA? We know the full form of DNA is deoxyribonucleic acid. So, what is DNA? It is a genetic material, and the genetic material which has instruction for making and operating an organism. That means what this DNA is actually a genetic material, and why it is called a genetic material, because it is having information or instruction for making and operating an organism. So what an organism does and what that organism will do, that all is being encoded in the forms of instruction on DNA. So DNA is divided into different sections, and that are called as genes. So actually gene is composed of DNA, and the certain sections of DNA are known as genes. So what is gene? So we have till now we have got an idea what are genes. So genes are part of DNA and carry hereditary information. So these genes carry hereditary information, and that information is passed from parent to progenies. That is, information is being passed from one generation to next generation with the help of gene, which is a segment of DNA. Then another related term is alleles. So what are alleles? Alleles are actually the alternate form of gene. So a gene may be in two or three format. So allele, the one or two or more forms of a gene are known as alleles, and it produces variation in a genetically inherited traits. So with the help of alleles, what alleles will do? It will create variation. Suppose a gene is characterizing a character or a trait. So one form of gene will signify means one form of trait. Another form of that particular gene, that is a different allele, will signify different trait. So here you can see in nutshell, you can see in a cell there is nucleus. In nucleus there will be chromosome. Chromosome will be form of DNA, and segments of DNA are known as genes. And in genes are alleles, and genes you can see here different form of gene. Here you can see it is a brownish color, and here it is a bluish color. So different form of gene is known as, or alternate form of gene is known as allele. So I hope now it is very clear to you what are genes and what are alleles. Actually many a times in genetics, whenever we are means we are studying Mendel's law or Hardy-Weinberg law, or thereafter many things. So in that case, many a times we are using genes or alleles as a synonymous. Actually, it is not a synonymous, but we can use genes or alleles alternatively or interchangeably. Now, the next term is locus. So what is locus? Locus is actually you can say in the literal meaning of locus is a house of gene. So what is house of gene? It is a particular place or location along a chromosome where gene resides. So it is a place where genes are located or alleles are located. It can be gene or other DNA sequences. So here you can see this is the locus where different form of genes are residing. So on a particular locus, there will be only one gene or allele. Another related term, actually there are two related terms. The one is genotype and another is phenotype. So these two terms we are facing frequently when we are studying animal genetics. So what is genotype? So genotype is genetic makeup of an organism. So actually an organism, the makeup, genetic makeup of an organism, that what genes it is possessing, what alleles that organism is having, that makeup of an organism is called as genotype, and actually this genotype will determine the phenotype. So the thing is that what is phenotype? So we know the genotype, now we know that genotype is a genetic makeup of an organism. So similarly, what is phenotype? The phenotype is appearance or characteristic of an organism. So outwardly expressed traits or externally appearance of an individual, in terms of what? An individual or an organism will express itself with certain traits. So external expression of traits or characteristic of an organism is known as phenotype. So this genotype and phenotype is actually related. Genotype is the genetic makeup of an individual, whereas phenotype is the outward expression or external expression of the genotype of an individual. It is called as phenotype. So next is the next here you can see another related terms is the dominant and recessive. This may be dominant gene or dominant genotype, or recessive gene or recessive genotype. Here in this particular situation, I am going to discuss about the dominant gene. So what is dominant gene? It is a dominant version of allele of a gene which shows its specific trait even if only one parent passed the gene to the progeny. That means if I am talking about dominant, there means you can think that okay, so another form is there which is known as recessive. So but the thing is that what is dominant? So dominant means it is a version of allele, the dominant version of allele. When an allele will be called as dominant, when only if it is being transferred from one parent only and it is expressing its characteristic. So once you will know the definition of recessive, then I think it will be more clearer. So once again I will read. The dominant gene means it is a dominant version of a gene which shows its specific trait even if only one parent passed the gene to the progeny. If only one, there are, I know, means we know that there are two parent, mother and father. So even if one parent is giving this version of allele to its progeny, in progeny this version is getting expressed or expressing its trait. So this is known as dominant gene. What is recessive gene? The recessive gene shows its specific trait when both parents pass the gene to the progeny. That means when both mother and father both are passing this version of allele to its progeny, then and then only the progeny will be able to express this character. So this is called as recessive. So I hope this dominant and recessive gene, the concept is clear to you. So what is dominant? When only one parent, even if one parent is transmitting it to progeny, the progeny will show this character. Whereas in case of recessive, when both the parents will transmit it to the progeny, then only the character will be expressed. This is called as recessive. Another related two terms are homozygous and heterozygous. So what are the homozygous? Homozygous individual possessing a pair of identical allele at a particular locus. So here in this picture you can see this, both the red, this is representing a gene or allele. So on both the chromosome is at both the homologous chromosome. Homologous chromosome is one chromosome from mother and another chromosome from father. So both these chromosomes is possessing similar alleles or identical alleles. So that is called as homozygous. Whereas when they are possessing dissimilar or unidentical, unlike genes, here you can see the mother is possessing pink allele and father is possessing blue allele. So these two unlike alleles on a particular locus is there, then this kind of individual is known as heterozygous individual. So this homozygous and heterozygous concept is very important when we will study in next few classes about the Mendel's law or exception to Mendelism. Then we will encounter this term more often. The next is the haploid and diploid. Another two related term. The haploid, it is the condition of having only one set of chromosome per cell. So if in a cell there is only one set of chromosome, so we know that chromosome there will be two set of chromosome. One set I will be suppose I am an individual, I will be in my each cell I might have received one set from my father and another set from my mother. So there may be cells in an organism where only one set of chromosomes are present. So such type of cells are known as haploid cells, or such condition is known as haploidy. Diploid cell. So what are the diploid cells? The condition of having two sets of chromosome per cell, 2n. So the cells which are having two sets of chromosome, one from mother and another from father. Here in this picture you can say there are three type of chromosome. You can see orange chromosome, purple chromosome and yellow chromosome. So in diploid you can see each chromosome is having two copy, that is one from mother and another is from father. Whereas in haploid you can see there will be only single copy, either from one parent. Now another related term is zygote. So what is zygote? Zygote is actually a diploid cell, and it results from the union of two gametes in sexual reproduction. So we know in sexual reproduction there will be fusion of male and female gamete. So after fusion of male and female gamete, you will get zygote, which will be having the characteristic of diploid cell, or it will have two sets of chromosome, one from male parent, another from female parent. Now another related term is chromatin and chromosome. So what is the difference between chromatin and chromosome? Actually the chromatin word, it's a complex of DNA, RNA and proteins. So there will be DNA, RNA and proteins. These three together will make the chromatin. Actually what these three will together form chromosome also. But the difference between chromosome and chromatin is that this chromatin will be the uncondensed eukaryotic chromosome. So we know at a particular stage of cell cycle, the chromatin material, they will start dehydration and they will be start condensation, and in condensation they will become thicker and thicker and they will appear like a chromosome. But in the resting stage of cell cycle, that you may call it as interphase. At interphase, the chromosome will be fully hydrated and they will be uncondensed. They will be spread, they will be under the Brownian movement inside the nucleus, and that structure in combination with DNA, RNA and protein will be known as chromatin. Now the second term is chromosome. So I have already discussed the structure within the nucleus of eukaryotic cell composed of chromatin and visible at cell division only. So at certain stage of cell cycle, maybe you can say it mitotic M phase, mitotic phase or meiotic phase. At that stage, the cell become condensed. This is the chromatin material, they become condensed, and after condensation they will form, they will known as chromosome. So the chromosome is actually a condensed chromatin. Now the related term is homologous chromosome. What is the meaning of homologous chromosome? Actually this also in due course of when I was discussing various other terms, I think I have given the concept of homologous chromosome. So what are the homologous chromosomes? The chromosomes that are similar in morphology. So they are similar in structure, they are similar in shape and form and genetic constitution. So these chromosomes, homologous chromosomes, they are similar in morphology and similar in genetic constitution also. In animals, one set come from the father and the other come from the mother. So homologous chromosomes are actually similar in morphology, similar in genotype or genetic makeup, and they might have come one from mother and another from father. So here you can see these are the two similar chromosomes. One has come from mother and another has come from father. So these two are the homologous chromosome, and one chromosome might have two chromatids. So another related term is chromatids. So you have to differentiate between the chromatin, chromatids, chromosomes. We know in the due course of time, when we will go deep inside the principle of genetics, it will be very pertinent if you know the differences between these related terms. If you are having any confusion in these related terms, it will be very difficult for you to understand the principle of animal genetics or genetic principles. So the next term is chromatids. So what are the chromatids? It is one of the two halves of duplicated chromosome. So as I have already shown you in the last figure that it is one of the two half of a duplicated chromosome. So when chromosome condense, it will get duplicated, and the one portion, one half will be called as chromatids. Another term is centromere. So we know the centromere. What are the centromere? These are the specialized constricted region of a chromatid. So in the last figure, if you remember, or if you not remember, I will just show you. This is the region. This is the region, the constricted region of a chromatid, and this constricted region is known as centromere, and it composed of kinetochore. So kinetochore is a protein. So what is this? This is the specialized constricted region of a chromatid that contain the kinetochore. Sister chromatids are joined at the centromere during cell division. So at the time of cell division, these two sister chromatids join together at a point. That point is known as centromere, and it is composed of kinetochore. Kinetochore is a protein. So when you will study about the cells and cell division, then you will study these things in greater detail. So we know that on the basis of position of centromere, we have classified chromosomes into different types, and different names has been given. So that we will study in the later classes. So another related term is recombination. What is recombination? So recombination is exchange of genetic material between the chromatids. So here you can see this is one chromatid, this is another chromatid, this is another chromatid, and this is another chromatid. So these are the two homologous chromosomes. And in this case, you can see that these two purple chromatids, these two blue chromatids, they are the sister chromatids. They are the sister chromatids. Again, these two green chromatids, they are the sister chromatids. But this green and purple or blue, this green and blue chromatids, if we consider these two chromatids, green and blue, they are non-sister chromatids. So the exchange of genetic material between the chromatids, and it often occurs between the non-sister chromatids. So this is called as recombination. Actually, recombination is the result, and the result comes by after a phenomena. So what is that phenomena? That we will see in the next slide. So the phenomena is crossing over or crossover. So the breaking and rejoining of homologous non-sister chromatids. So as I have already discussed the non-sister chromatids and homologous chromosome, that means the one set come from mother and another set come from father. So the breaking and rejoining of homologous non-sister chromatid during early prophase 1 of meiosis resulting in recombination. So crossing over, what is crossing over? It is a breaking and rejoining of homologous non-sister chromatids during early prophase 1 of meiosis, and it will result in recombination. So this is about the crossing over and recombination. Another related term is synapsis. So what is synapsis? Synapsis, the term itself tell synapsis, the literal meaning is pairing. So the pairing of homologous chromosome, during prophase 1 of meiosis is known as synapsis. So here you can see these two are the homologous chromosome, and this is the pairing of homologous chromosome. So this is called as synapsis. And you will study when I will be telling you about the meiosis or meiosis. Then at that time I will tell you that at that time of formation of synapsis, there will be formation of synaptonymal complex, a protein complex. So synaptonymal complex will be formed between the synapsed chromosome. So after that, this synaptonymal complex will facilitate the pairing of chromosome, and thereafter the crossing over and recombination. So this is the product after the crossing over. So this is about the synapsis. Then the similar term is not similar but related term is the disjunction. Disjunction, the name itself tell that there will be breakage of junction. So what is breakage of junction? It is a separation of homologous chromosome during anaphase. So at metaphase there will be pairing of chromosome, there will be formation of junction, and these sister chromatids, they are joined by centromere at a point. And during anaphase of cell cycle, anaphase stage of cell cycle, these two sister chromatids will be separating with each other, and that is called as disjunction. The disjunction is very important event, and in later classes you come to know why it is important. Another related term with disjunction is non-disjunction. So when we will see the phenomena of linkage and crossing over, we will study about the phenomena of linkage and crossing over. We will again come encounter with this term, the non-disjunction. At that time we will discuss what is non-disjunction. Now another term is parent generation or parental generation. So what is the parental generation? So when we will study about the Mendel's law or other principle of genetics, so parental generation will come many times. So what is parental generation? The generation that supplies gametes to the filial generation. So parental generation will supply gametes to the filial generation, means coming generation, next generation, or progeny generation. So what is filial generation, or it is called as F dash or F prime? The generation that receives gametes from the parental generation. So the generation that receive gamete from parental generation is called as filial generation. There will be first filial generation, second filial generation, and so on and so forth. So here in this figure you can say this is the parental generation. It is having two type of gamete, capital P and small p, capital P and small p. And these two gametes, they will be crossed in the next generation. The first filial generation, they will form the hybrid capital P and small p. And another, if we cross capital P with capital P small p with capital P small p, there will be formation of second filial generation. That we will see when we will be studying the Mendel's law. So for right now, you have to remember that parental generation are the generation which will give gamete to the filial generation, and filial generation is generation that will receive gamete from the parental generation. Another term is hybrid, an offspring resulting from mating between individuals of two different genetic constitution. So when individuals of two different genetic constitution, here you can see the capital P is one kind of genotype, and small p is another kind of genotype. So when capital P will be crossed with small p, they will form hybrid, that is capital P and small p. So these first filial generation individuals are known as hybrid. Then based on this hybrid, there are two related terms, and similarly there will be many related terms also, but right now here we are discussing two terms, the monohybrid cross and dihybrid cross. So what is monohybrid cross? A genetic cross, we have already seen in the last slide. It is a genetic cross that takes into account the effect of alleles at a single locus. So we are considering single locus only at a time. So we are seeing the effect of alleles at single locus, or you can better you say it's a single gene. So this is called as monohybrid cross. Similarly, the dihybrid cross is a genetic cross that takes into account the effect of alleles at two separate loci. That is two different genes. So when two genes are under consideration, it is called a dihybrid cross. When one gene is under consideration, it is called a monohybrid cross, and similarly trihybrid, tetrahybrid, and so on and so forth. Okay, so this is the about the monohybrid and dihybrid cross. Another term is gene pool. So this is very common term. What is gene pool? All of the genes in all of the individuals in a breeding population. So suppose you are having a breeding population. So all the genes that are being possessed by all the individuals of that breeding population are called as gene pool. So more precisely, you can say it is the collective genotype of a population. So whatever population, what population you are having, all the genes that is being possessed by that population is known as gene pool. So this is the monohybrid cross. Here you can see only one gene is under consideration, that is A gene. A gene is having two alleles, capital A, small a. Similarly, if you see the dihybrid cross, this is called as monohybrid cross. This is dihybrid cross. Here two genes are under consideration, A and B. So A is having two alleles, capital A, small a. B is having two alleles, capital B and small b. So this is dihybrid cross, this is monohybrid cross. Next, after seeing all this term, the one term that combines all the terms is genetics. So what is genetics? Actually, in simple term, you can say the genetics is the study of heredity and variation. What is heredity? Means transmission of characteristics from one generation to next generation, or transmission of gene from one generation to next generation. And if genes is getting transmitted from one generation to next generation, it will bring uniformity. It will bring uniformity, means all the organism, parents and progeny will be uniform. But there are variation also. So how these variations are being created? How one gene is getting transmitted to next generation? This all we are studying in the genetics. So what is genetics? It's the study of gene structure and action and pattern of inheritance. So we are studying the structure, action and pattern of inheritance of trait from parent to offspring. It is the branch of science that deals with the inheritance of biological characteristics. So not only inheritance, but we are studying the variation also. So how this variation is being created? If a gene is as such getting transmitted to next generation, then everything should have been uniform. But it is not like that. we know that no two organisms in this world are same. There are variation. So how this variation are getting created, this also we are studying in the genetics. So genetics is the study of heredity and variation. Another term related with this is genome. So what is genome? Genome is the full genetic full genetic complement of an individual or of a species. So what is genome? Genome is the full genetic complement of an individual or a species. So whatever genetic material, genetic complement an individual or a species is possessing, that is called as genome. So in human, it is estimated that each individual possesses approximately 2.9 billion base unit in his or her DNA. So what is base? So we know the DNA is composed of deoxyribonucleic acid, and it is composed of sugar, phosphate and nucleotide base. And these bases are the building block of DNA, and they are the major one who are creating differences between the different individual or similarity between the individual. And actually the information is being stored in these bases, their arrangement, their sequences, etc. So in a human being, there are approximately 2.9 billion base units. Similarly, it may be varying in other organism and other species also, but the difference is not that much, but still there are differences. So we will study in the next coming classes about the principles of animal genetics, then different related terms, related phenomena, the Mendel's law, Hardy-Weinberg law, exception to Mendel's law, exception to Hardy-Weinberg law, and many more things. So for as of now, I think this is enough for the terms related with animal genetics, and if any terms is remaining, that we will discuss as we will discuss the different principles of animal genetics, and when those terms will appear, we will discuss those terms at that point of time. So thank you very much.